Hello friends, today we are going to talk about antimicrobials, particularly antibiotics. Now we all know many infectious diseases are caused by different microorganisms, particularly bacteria. To treat such infections, we use specific chemotherapeutic agents like antibiotics, which simply are the substances or compounds produced by one kind of microorganism to inhibit or kill other bacteria, microbes present around it. So the property of an antibiotic is it inhibit or either kill a pathogen while harming host as little as possible. So it either inhibits or kills the pathogen. Inhibition is simply means stopping the growth. That means if you remove antibiotic from the system, microorganism may regain their growth. So it is basically giving a chance to our immune system to take over the infection. While killing the pathogen is simply means destruction of that microorganism. As far as types of such antibiotics are concerned, as I said, they target specific microbial processes which are not present in host, the eukaryotic host. So they damage pathogens by different mechanisms. The first mechanism is inhibition of cell wall synthesis. Peptidoglycan, you all know prokaryotic prokaryotes possess a unique cell wall, that is peptidoglycan, which is not present in eukaryotes. So this is a good target to work on. Next is inhibition of protein synthesis. Very important process. Generation of proteins is a crucial step in the metabolism of any organism. So we target that. In third case, we have nucleic acids. You all know the significance of nucleic acid to any organism. The next one is disrupting of microbial membrane structure and function. Again, membrane is very crucial. The next to the cell wall, there is a membrane, and various transport and energy generating mechanisms are associated with the membrane structure and function. So it's a very important structure for a cell's life is concerned. Next, blocking the metabolic pathways through inhibition of key enzymes. There are specific enzymes which are playing a very crucial role in the cell's metabolism. We target only those specific enzymes of a particular pathway. This is an overview of all the drugs, representative drugs of specific family and their mechanism of action. So first we will talk about the cell wall. So those antibiotics which block the peptidoglycan synthesis and also repair of whatever damage is happening to the cell wall. So penicillin, cephalosporins, vancomycin, bacitracin and monobactams. Monobactams are recently discovered. Next we have protein synthesis inhibitors, so drugs which act on ribosomes and they somehow block the protein synthesis mechanism. So some antibiotics bind to the 50S subunit. You all know prokaryote possess 70S ribosomes, Svedberg unit. So there is 50S subunit and 30S subunit. So chloramphenicol, erythromycin, clindamycin, they go and bind to 50S subunit, blocking the ribosome function. While some antibiotics like aminoglycosides, tetracycline, streptomycin and amikacin, they interact with 30S and block the protein synthesis process. Next is DNA synthesis inhibitors. So DNA replication, all the enzymes present in the process of DNA replication, supercoiling of the DNA, all these are specific to prokaryotes. So these is a good target for an antibiotic to development. So the enzymes include DNA replication enzymes, transcription, so mRNA synthesis is blocked. So examples of enzymes are gyrases, RNA polymerases. So gyrases are inhibited by quinolones, ciprofloxacin, levofloxacin, and rifampin, which is a classical drug to treat tuberculosis, inhibits RNA polymerase. The next one is cell membrane. The antibiotic which interact with the cell membrane is polymyxin. So there are peptide antibiotics which basically interact with the membrane and create pores in the membrane, and therefore we have osmotic lysis of the cell. The next part is cytoplasm. So there are specific enzymes, specific metabolic reactions are targeted by some antibiotics. For example, sulfonamides. Earlier they are developed to treat diabetes, but accidentally they were discovered which were hampering the specific process in bacteria. So here they act as a metabolic analog and participate in a particular reaction blocking the enzyme activity. So trimethoprim and sulfonamides are the two antibiotics which inhibits the precursor formation which are required for DNA synthesis. Now, what are the characteristics of an ideal antimicrobial agent? The very first property, which is true for any therapeutic agent, not just antibiotics, is selective toxicity. Means it must kill or inhibit the microbial pathogen while there should not be any damage to host. That means whatever the process it targets, it should be targeted only those processes which are unique to microorganisms, prokaryotes, not eukaryotes. Second thing is larger therapeutic index. The simple way of explaining this term is the body should tolerate more and more amounts of a particular substance while it should be active at very low concentration. That simply means. So basically therapeutic index is a ratio of toxic dose to the therapeutic dose. It should be either cedal or static. So cedal means complete destruction of the microorganisms. So membranes will be ruptured, cell wall will be ruptured, etc., etc. And stasis means only the growth is inhibited so that immune system is there counteracting in that case. So immune system will take care of infection. So either it can be cedal or static. Some antibiotics can be narrow spectrum or broad spectrum. So narrow spectrum means, say example penicillin, which only targets gram-positive organisms. However, gram-negative organisms are not affected. But some higher penicillins, piperacillin, tazobactam, all these antibiotics are active against gram-positive as well as gram-negative organisms. Second most important thing is do not allow the development of antimicrobial resistance. The given drug should not allow development of antimicrobial resistance, which is most important public health concern today. Next, it should complement and assist the activities of host defenses. So first thing we have already discussed that it should not damage host. So if it is not doing that, it's okay. But if it assists immune system of host, it will be an added advantage. It should remain active in tissues and body fluids for long periods of time. It should have a good distribution. So we have to do pharmacokinetic studies. Should possess a good pharmacokinetic profile. Readily delivered to the site of infection. It's very easy, or it should be easy to reach out all the parts of the body and should be introduced very easily in the body. Most importantly, it should not be excessive in cost, so less cost, and does not disrupt host health by causing any allergies, because we all know that around 10% US citizens are allergic to penicillin, so we have to prescribe cephalosporins to them. So it should not create any allergy in the patient. So obviously we have to do a lot of clinical trials before bringing that antibiotic into the market. Thank you. Stay tuned for the next video.